few examples. In Parkinson, what is used is a molecule called 6-hydroxydopamine. If you inject it in a specific region of the brain called the substantia nigra, it kills the cells making dopamine, and that's exactly what we have in humans with Parkinson. Those dopamine neurons in that region of the brain die. So you inject 6-hydroxydopamine and the animal will develop Parkinson. The way that we quantify Parkinson in a mouse, which is essentially a lack of coordination, is that normally a mouse will walk. Remember, you've all seen a mouse somewhere. It walks, it stops, smells a little bit, it walks, it stops, it walks, it stops. It will stop on average 10 seconds when it stops. Its time of stillness is about 10 seconds. When it develops Parkinson, it will go above a minute. The mouse is reluctant to move because of the lack of coordination. If you give the mouse L-DOPA and carbidopa, the typical treatment that is given to humans, you can see an improvement. But if you trigger the mouse own stem cell, the release of the mouse own stem cell, you can see a condition that is almost back to control level. Now, in that study, they wanted to quantify the actual evolution of the loss of coordination. So what they did is that they put on the nose of the mouse, they put a piece of tape. And they counted the time that it takes for the mouse to remove the tape. Scientists sometimes are creative. So you just need something that you can quantify. So they did this test 1 day after the injection of 6-hydroxydopamine, 7 days, and then 2. So 1 day, 1 week, 2 weeks. You can see the control group is actually a learning that takes place, but in the group that is developing Parkinson, you can see the evolution of the loss of coordination. It takes, 25 seconds after 2 weeks to remove that tape. You give L-DOPA, carbidopa, you have the same trend, it's just slower. You trigger the release of the mouse own stem cell, you get the same damage, and then repair. And we have seen the same thing in humans. This has been documented in humans. So I'm just showing you here 3 different models of health problems where stem cells can really contribute to tissue repair. Similar data you can find for spinal cord injury, for liver problems of the lung. So at the end, the conclusion is simply that if you can increase the number of stem cells in circulation, forget now diseases. We need to use them to show the phenomenon. The conclusion is simply, if we can put more stem cells in circulation, the body can use these stem cells to simply repair itself. But all of this is leading to a third conclusion, which to me is probably the most important. That's the one that, when I say it's called to change the way that we understand health and wellness and practice medicine, this is what I mean. It's the corollary of these two. More stem cells in circulation means that you can better repair. Fewer stem cells in circulation means that you are at risk for developing problems. And here's what this means. Today in the world overall, in the medical world, I mean worldwide, the understanding of disease formation, we don't talk about it like this. Because we look at diseases as labels. You have Parkinson, diabetes. These are all labels with their set of symptoms. But if we really look at it carefully, every single degenerative disease is cellular loss in a specific tissue. You lose cells in the pancreas, you've got diabetes. You lose cells making dopamine in the brain, Parkinson. You lose cells making T3, T4, hypothyroidism. You lose cells making the Schwann cells around your neurons, multiple sclerosis. You lose cells making acetylcholine in the brain, Alzheimer's. Think of any degenerative disease. It is always the loss of a type of cells. And the general concept is that you're born, you mature, you reach 20, 25 years old, you have peak health, and then from there on, everything is a slow decline. Isn't it the way that we look at health? And then wherever is your weakness, because of your genetics, because of many factors in your life, you will be exposed to develop a disease more than another. But all this science on stem cells is telling us that that is not what is happening. And it's studies like the one that I talked about before, the one on leukemia, where you irradiate female mice, you inject male stem cells to look at the reconstitution of the bone marrow in the blood, and we found a brain cell with the Y chromosome. These mice did not have brain injury. So while stem cells will migrate specifically where you've got an injury, they also migrate to a lesser extent to many different tissues constantly to replace the cells that are being lost. In other words, and if you think about it, it's so logical. Summer is not all the time. It goes to fall, it goes to winter. Everything is cyclic in nature. Your body is cyclic as well. You lose cells every day, you replace them, everything is in a process of turnover. There's one study that is studying that more specifically, so let's look at it. It's a cool study. It's a study that used a phenomenon in nature, which is the fact that the level of radioactive carbon in the atmosphere was essentially zero until we started to do nuclear bomb testing on this planet, and then the concentration of radioactive carbon started to rise very, very sharply until 1965, when there was a ban on above-ground nuclear testing. From then on, radioactive carbon started to dissipate into the stratosphere and slowly started to disappear. Now, the carbon that is in the atmosphere will be incorporated through photosynthesis into plants that we will eat, or that animal will eat, and we will eat the animals. So when a cell in my body divides, then I need to divide the DNA. I need to make a new filament of DNA. These filaments of DNA are made out of carbon. So when I make a new filament of carbon, where is that carbon coming from? It's coming from my food. Where's my food coming from? It's coming from the environment. So the ratio of radioactive carbon that is in my food reflect the ratio of radioactive carbon in the atmosphere. So the ratio of radioactive carbon in my DNA filament will reflect the ratio that was in the air in the months around the time that that cell divided. So that means when I look at this, they did that study in heart cells. So if I take heart cells from somebody born after 1965 and I quantify the ratio of radioactive carbon in the filament of these heart cells, I can see two things. Either all the radioactive carbon, the ratio reflects the 3 to 5 years of age at the time that the tissue matures, and then after that I have no new cells. Or I can see that during the entire life of the individuals, I see new cells appearing all the time. And that's basically what was seen. So you basically quantify the radioactive carbon, and it tells you the date that a cell was born. And the conclusion from that study, we have half of a new heart every 25 years. Depending on studies, I will see you've got a new heart anywhere between every 30 to 50 years. Other studies seem to indicate that it could be more rapid than this. But generally speaking, the conclusion is that you've got a new liver every 2-3 years, a new pancreas 4 or 5 years, new lung 4 years, new muscles 9 years, new skin every week or 2, new brain, there's hope, it replaces as well. There's no clear data as to how fast it goes, but we also regenerate the brain. Keep in mind, my background is neurophysiology, brain research. I was told not brain research, but in my studies, in my schooling, I was told the brain does not regenerate. That's what I'm sure you've learned and you've read many places. Not true. The brain actually regenerates a lot. Okay? So the conclusion is that we are constantly in a process of tissue renewal. So in reality, health is not a slow decline. Health is a balance between two processes in the body. On one end, you lose cells every day, but you replace them also every day. When you're young, you've got a lot of stem cells in your body because stem cells are made by the red marrow, and as you age, the red marrow transforms into yellow marrow. So as you age, you've got fewer and fewer stem cells. So there's a point in your life where that balance shifts, and now you no longer have enough cells to compensate for cellular loss. That's when you start to be in trouble. You're not yet in trouble. It's like, a good analogy here would be like your bank account. The previous worldview is that you're born with a set amount of money in your bank account, spend it wisely. We discovered that it's not the case. You've got a regular income, and as much as it matches your expenses, you're fine. The day that your income declines, then that's when you start to be in trouble. You're not bankrupt yet, but you start to be in trouble. The day will come and you'll be bankrupt, and that's the disease. And what will make you develop a problem or another, it's a whole slew of factors that you all know very well. Your genetics, your past injuries, environmental toxin, exposure to environmental toxins, your lifestyle, your exposure to physical stress, emotional stress, all of these, your mother-in-law, put all of this in the bag. It's the pressure that is applied on you and your ability to deal with it. I mean, what I'm saying here is something that you've all seen in your life. Two people with the same problem, one of them recovers quickly, deals with it, the other one is crushed by it. Haven't we seen that in life?
Yeah.
All of these are different. So that will determine why somebody will develop Parkinson's, somebody else it's diabetes, somebody else needs a new knee. So we're all different. Let me take again the banking analogy. It's like saying you've got someone who cannot pay for their car, somebody else cannot pay for their house, mortgage problem, somebody else cannot pay for their study, somebody else cannot pay for their health bill. So we've got health problem, school problem, mortgage problem, and car problem. But don't we really have only money problems? We've only got money problems. But in different people, it shows up in a different way, depending on the context and the circumstances, and we call it differently. It's the same thing. The baseline fundamental problem is an inability to properly compensate for cellular loss. And that loss will show up in whatever is your weakness, and it will have a different label. You get it?
Yeah.
Now, if that is true, because 8 years ago, 10 years ago, it was an idea. It was just an idea. I'll tell you where this idea came from. Just opening a parenthesis.